Hello.
Hello.
കേക്കാവോ?
കേൾക്കാം.
Okay.
പുള്ളി നമ്മുടെ സ്പീക്കർ വന്നായിരുന്നു
സാർ.
വന്നായിരുന്നല്ലേ?
ഡോക്ടർ അരുണനാണ്.
Maybe my ID is something like Scripps, no? That's why I log in.
Yes, sir. Hi.
Sorry. Hello.
Hello.
Yes, കേൾക്കാം.
സാറ് പറഞ്ഞോളൂ സാർ. അരുണൻ സാർ.
You can introduce.
സാർ ഇൻട്രൊഡ്യൂസ് ചെയ്തോളൂ.
No, you can introduce. No, I will at the end I will manage. Sharika, you can introduce.
Okay, sure, sir.
Hello.
Hello.
Yeah, can I start?
No, we'll just introduce and after that you can start.
Okay. All right.
It's 11.
It's 11.
Okay, fine. Arun sir?
No.
Shall we start?
Yeah, we can.
Okay, fine. So, good morning all the participants. Today we are having the third webinar of the series on the biotechnology education, the challenges and possibilities. And for this day we'll be looking on to biotechnology with a focus on nanotechnology. And for that we have Dr. Binulal Sathy from Amritha Center for Nanoscience and Molecular Medicine. Let me just introduce the speaker. Dr. Binulal Sathy now serves as the Assistant Professor at the Amritha Center for Nanoscience and Molecular Medicine, Amritha Vishwavidyapitham at the Kochi campus. And he's the recipient of the prestigious Ramalingaswami Fellowship from the Department of Biotechnology, Government of India. And he started his career as a research assistant at the Institute of Biomedical Sciences, Academia Sinica, Taiwan, in the area of medical biotechnology. He obtained his PhD in bioengineering from the Amritha Vishwavidyapitham, following which he did his postdoctoral work at Nico's lab at Rice University, USA, and Kelly Lab at Center for Bioengineering, Trinity College, Dublin. And after his postdoc, he joined as a member of faculty at Amritha Center for Nanoscience and Molecular Medicine in 2017. And his research interest is in the area of regenerative medicine, with special emphasis on investigating the immunomodulatory and regenerative properties of adult stem cells and biomaterials and their bioengineered constructs. Developing bioengineered niches for achieving self-renewal of hematopoietic stem cells ex vivo is another major focus of his research. He has over 30 peer-reviewed research publications and 2 big chapters to his credit. And on behalf of Kerala Biotechnology Commission and Kerala State Council for Science, Technology and Environment, let me heartily welcome the speaker, and over to you.
Thank you, Sharika.
Thank you.
Yeah. Hi, good morning to all. Hope all of you are staying safe. And I can start, right, Sarika? I can just present.
Yes, you can start right now. Yes.
Yeah. Give me one quick second.
You can share the slide.
Yeah, one second. Hello? Can you see
Hello.
my slides?
Yeah. It's
Just coming?
yep. Yes.
Yes, now it's visible. You can make it
Full screen. Make it.
It's full screen?
Yeah.
Yes.
Okay, fine.
And then you can hide that. Here, stop sharing and hide.
Sorry for the mess. So,
No problem.
sharing. I mean, because I can't see it. Give me a second.
Yeah, now it's visible.
It's visible. All right.
Yeah.
Okay.
Not at the full screen.
Give me a second. Now, how about now?
Yeah, fine.
All right. Okay.
Yeah.
Okay. Good morning, all. As introduced by Dr. Sharika, myself, Binulal and Sathy. And our today's discussion is on emerging prospects in biotechnology and the impact of nanotechnology. If I understand it correctly, I'm talking to a group of young biotechnologists or biologists in the related fields who are going to be the pioneers in the field of biotechnology and nanotechnology in the coming days. I'm so happy to talk to you because I also started as a biotechnologist. I started my research career after obtaining my master's in biotechnology. And currently I serve as a faculty member here in Amritha Center for Nanoscience and Molecular Medicine. And a couple of words about my center. As you can see in the screen, so we are in the healthcare campus. We are located in the healthcare campus. Amritha Institute of Medical Sciences is a multi-specialty hospital, and our research center is associated with it, which I have shown with an arrow. Our center is the first nano bio center with research and academic components. And the motto of our university is Shraddhavan Labhate Jnanam. The earnest aspirant gains supreme wisdom. And we believe in that, and we train our students according to that. And this slide shows the thrust research interest of our center. The first is nanobiotechnology and bioengineering, wherein tissue engineering and stem cell research is one of the major aspects, and cancer diagnostics and therapy, nanomedicines, biomedical implants and nanotoxicology, and much more. And second one may not be an interesting topic to us, but it is nanotechnology and renewable energy, which talks about the solar cells, etc. And the other area is molecular medicine, which is the basic and applied aspects of molecular medicine, molecular and cellular phenomena, disease prevention, in the molecular aspects. See, the bottom line is enabling high quality products in various scientific disciplines using nanotechnology. So let's come back to our topic. it's biotechnology, right? So we all agree that biotechnology is a frontline area of science with immense potential for the benefit of the humankind. So today, in 2020, we are all fighting against the pandemic in all possible ways. And we know that there is extensive research happening in the area of biotechnology, that is in the vaccine development against this COVID-19. And you may be surprised, I think all the listeners would be aware that now the vaccine is the latest advances in the vaccine area is in the mRNA vaccine, which is a nano vaccine. So in addition to the nano vaccine, the traditional vaccine technology is currently in fruit to fight against this COVID-19. But this term, this biotechnology, was coined by a Hungarian engineer called Erki in 1919, that is 100 years now. At that time, it was used for the purpose of living organisms and for the production of new products from raw materials of biological origin. So these biotechnologies, including us, we are experts in genetic, chemical, physical attributes of cells, tissues, and organisms in order to develop new technologies, processes, and products that will improve the quality of human life. This is important. So we are experts. We are supposed to be experts in the genetic, chemical, and physical attributes of cells, tissues, and organisms in order to develop the new technologies, process, and products that will improve the quality of human life. So we know the various aspects of biotechnology, such as environmental biotechnology, medical or healthcare biotechnology, industrial biotechnology, agricultural biotechnology, biofuel biotechnology, and marine and aquatic biotechnology. These are various streams. It's a busy slide, but I'm not going to the details of these. Because we are all from the related areas, we know this all depends on the individual choices, which area we want to focus. All these areas are equally important, and they are and they need contribution from the young folks in the biotechnology. In all these areas, our country needs it for a sustainable development of our country. So in this one, I'll be focusing more into the medical and health biotechnology area. And as I just showed here, the ground is ready and it is open for all of you. Which area you want to contribute, you can decide based on your interest and your knowledge and your expertise. So let's have a quick look at the biotechnology industry in India. So biotechnology industry in India, or biotechnology in India, started with the establishment of the Department of Biotechnology in 1980. And in 2003, they had an investment of 1.1 billion US dollars, which grown into billion industry in 2018. And now we should be happy to say that India is among the top 12 destinations for the biotechnology in the world. And we are the third largest biotech destination in the Asia Pacific region. And there are 2,500 plus biotech companies, 2,700 plus biotech startups. And they are expected to grow up to 10,000 by the year 2024. And we are the leader in the global supply of DPT, BCG, and measles vaccines. And we have the highest number of US FDA approved plants. We have around 665 outside the US and over 1,400 manufacturing plants, combined with the World Health Organization. And by 2025, we are expected to reach nearly 150 billion contribution to the global biotechnology market. So all these areas which I just showed in this slide can be categorized in these five areas. That is bioagriculture, bioindustrials, bio-IT, bioservices, and biopharmaceuticals. And the Department of Biotechnology, which is the leading agency which is controlling the biotechnology in our country, has set an ambitious target of the industry to make a US dollar 100 billion by 2025. That's a happy news to us, all the young aspirants of biotechnologies. So I titled this slide as "The Nation and the Industry is Waiting for Your Contribution." It's your decision where you want to contribute. The first is bio-based sustainable production of chemicals, energy, fuels, and materials using carbon dioxide as a raw material, seawater-based bioprocesses, biotech improvements to soil and water, accurate, fast, cheap, and personalized diagnostics and prognostics, rapid and precise development and manufacturing of medicine, zero waste food storage, engineering sustainable food production, advanced healthcare through genome sequencing, regenerative medicine, and non-resource draining of zero waste bioprocessing. So I want to spend one more second in this slide because all the listeners has to be, I would request you to remember these areas. So in the next phase of my presentation, I'll be talking about how nanotechnology may be helpful to you to use to improve your contributions, your research interests in any of these areas. I have listed the areas which is known to me. There may be more areas into this one, but if you are, what is more important is which area you want to contribute, which area you want to focus for your career development, which area you want to focus to contribute to the country. Okay, so now comes to the nanotechnology. So this is in the biotechnology area, we are focusing in contributing or producing products or educating people in all these areas. There comes this the terminology, the nano. So it is by this famous scientist, Richard Feynman, and he proposed that there is plenty of space. This is in one of his talks in the US. He's a theoretical physicist. In 1959 itself, he was talking about manipulating things or engineering things at the atomic, subatomic level. There comes the nanotechnology. So I titled it as Nano, the small giant of possibilities and potentials. Let's see what it is. I'm welcoming all of you to the world of nanotechnology. So I have two pictures here. You can in the right hand side, you can see an adult to an atom, where from the 1 meters to 0.1 nanometer. In the left hand side, you can see from a tennis ball to an atom. A tennis ball, this is in nanometers, okay? That is nearly 100 mm, and whereas the carbon atom is nearly 0.1 mm, 1 nanometer, sorry. So in this pandemic, time, we have to be very careful, and we have to know that what's the size dimensions which we are talking about in terms of a virus. It's coming around 100 nanometer. Just want to mention you, we were trying to develop a nano fiber-based mask system in our center. So we were trying to have a pore size which is much smaller than 100 nanometers. Now the research is focusing on, is there any possibility that we can capture these 100 nanometer particles, these viral particles, into the mask so that around in the environment so that we can destroy it. So I'm just telling you the size scale differences here and putting it in a much simpler way. An apple in the nanometer size, it is 10 to the power 8, whereas our virus is 10 to the power 2. Then comes the DNA, nearly 1 nanometer, water molecules 10 raised to -1. Okay. So I'm just showing a couple of slides for you to have a basic understanding about what we are talking about. Let's zoom into this picture. There is a small red, box there. Let's see, that's in 1 meter scale. When we go to a 100 millimeter scale, this is what we can see. When we go to a 10 millimeter scale, this is what we can see. When we go to further zooming, we can see that we are seeing much more details. And see, when we are moving to a micron scale, this is what we can see. This is a nucleus. When it is 100 nanometers, you are starting to see your chromosomes. And then 10 nanometers, you are starting to see your DNA. And then, of course, in 1 nanometer, you are starting to see the atomic molecules. So this nanotechnology is enabling us to see things in a much detail, so that we can apply the necessary modification which we want, if it is required, into the nanoscale. So let's see. So this is nature's beauty. I'm just pictorially representing that a person is just watching a butterfly. This is how general public will be seeing a butterfly, or an environmentalist would be seeing a butterfly. But in the eyes of a biotechnologist and a nanobiotechnologist or a nanotechnologist, he would be trying to see how nature made this beautiful creature into the nanoscale dimensions. See, its wings under a scanning electron microscope, you can see how it is, and it's further, zooming into the wing of this butterfly. You can see that much more detail, much more detail, much more detail. I can see that in the nanoscale dimensions, how beautifully it is orchestrated, this protein, particles there, the fibers there. And he might be thinking that how this can be, this kind of an array, this kind of an arrangement can be used for the benefit of the humankind. It can be providing a surface with significantly higher surface area for developing a biosensor. If we can develop a material like this, which can be conjugated with an antibody, which can function as a sensor. Another one, we would be familiar with this one, a caterpillar. See, if you take this one and take one of this hair, which is so itchy to us, if you take it to a SEM, and this is what you see. You can see those nano needles. See, that's the reason. These nano needles, this is one fiber. When you are seeing it in a SEM, in each needle, you can see multiple needlets. Multiple nano needles are coming out from this one needle. That's why when we touch, these nano needles are going into the skin, the pores of our skin, and that is causing us irritation. So can we have something like this one developed so that we can have multiple nano needles for injection, drug delivery, transdermal delivery? This is how a nanotechnologist or a nanobiotechnologist will think. Have you ever thought about why, when a mosquito is injecting or putting its needle into our body, we are not having pain? We may be getting irritation, itching. It is not because it is piercing our skin. It is because of that particular anticoagulant which it is pushing to our bloodstream to prevent its coagulation. But when it is putting its needle into our skin, it's not paining to us. So think about whether we can come out with something where even the small kids or even sustained delivery of drugs through our skin is possible by nanotechnology. I'm just giving you certain thoughts so that you would be able to think in the context of the area which you are interested in applying this technology. So the other one is, we are just putting a piece of hair. A piece of hair. A human hair is nearly 50,000 to 80,000 nanometers wide. That is, and it grows approximately 10 nanometers every second. Remember that. So now we are trying to focus, we are trying to zoom in much more. This is how you see your own hair, much more detail. I'm not very sure how clearly you can see this one, but below that you are seeing certain things. Okay, and here you can see the hair, the in one corner, and you can see small structures underneath. And if you zoom in further, you can again see those fibers, and these are the fibers. So now you compare how small we are talking about. Now in our center, we are producing these nanofibers continuously for multiple applications. Because in our human body, cells are interacting with dimensions not in the macro scale or micro scale. It is in the nanoscale. So when we are giving these fibers to our own cells, especially for regenerative medicine or tissue engineering, they are responding in a different way. So now just have a quick thought about, you all know the structure of DNA and its backbone and its the nitrogen bases, etc. So in terms of dimensions, the dimension of a DNA molecule, the diameter is nearly 20 Armstrong. 10 Armstrong is one nanometer, right? And one turn is nearly 10 base pairs is approximately 34 Armstrongs. So the bottom line is, all the genetic instructions for the development and functions of living cells are stored in DNA structure within the nanometer scale range. It's amazing, right? the old nanotechnology, I mean, is it something which we started very recently? No, it started long back. The first nanotechnologists, they identified that these gold nanoparticles, based on the size and its shape, they can display multiple colors. These glass paintings which you can see is based on the nanotechnology. At that time, they were not aware that this is nanotechnology, that's all. Because it's the nanoparticles of gold coated on the glass, which is in different size and dimensions, so that when the light hits in this one, it is reflecting different colors. So now what exactly it is, nanotechnology? So if you want to say a definition, it's the creation of functional materials, devices and systems through the control of matter in dimensions in the nanometer scale length. That is, even though below 1 micron, that is 1000 nanometer is nanoscale, we would like to say that the real nanoscale effect we can observe size scale is nearly 1 to 100 nanometer, wherein new functionalities and properties of matter are observed and harnessed for a broad range of applications. So it's uncommon, going small for big advances. So just another example of what I just showed you. So this is what light scattering of nanocrystals. So what's the effect of shape, size, and composition? See, it's the golden nanoparticles as prisms, sphere of different sizes. And you can see when the same light hits in these nanoparticles, it is emitting different color. So based on this principle, a lot of quantum dots are currently being developed, which is for the use of detection, early detection of cancer, etc. So this is again the production of these nanoparticles as different size scale, and you can see the color change here. So let's see what's the exact nanoscale effect in terms of biology. Okay, at the nanoscale, materials begin to demonstrate entirely new chemical and physical properties, such as materials can be stronger, lighter, highly soluble, have lower melting point, higher magnetization, higher conductivity, etc. So here I have 4 figures here. The first 2, on the left-hand corner, you can see something is hanging like a spider. So that is nothing but it's a synthetic polymer, which is a polycaprolactone, which is made as fibers on the microscale fibers as well as nanoscale fibers in the same platform. And you can see when you put human mesenchymal stem cells into this platform, how the cell respond. They preferentially like to attach the nanofibers, and they try to spread out, and they want to behave the way which it want. Whereas in the bottom line, you can see the same cell, same kind of cell, which is seeded onto a microscale fiber. And they like to sit in a round conformation, round morphology. Accordingly, its functionality will also change, right? On the right-hand side, so that's, I'm trying to say how the cells, individual cells are responding to the topography, the size scale. On the right-hand side, you can see titanium dioxide, the crystals, as well as nanopores of titanium. These are all possible things people are using for multiple applications. So the bottom line, which I want to tell you is, the smart use of nanotechnology can significantly benefit biotechnology industry to develop new technologies, processes, and products that will improve the quality of human life. So what exactly this nanoscience can do to biotechnology? Okay, hope you remember the slide which I have shown previously when I was explaining the biotechnology or my thoughts on biotechnology. So all these areas, whichever area you choose, environmental biotechnology or medical biotechnology, industrial, agricultural, biofuel, marine, whatever you choose, using this cutting-edge technology, you can improve the efficiency of whatever you are trying to develop. Okay, so the ground is ready and it's open for all of you. So it's a latest cutting-edge technology which can be used for the benefit of improving the outcome of all these fundamental biotechnology applications. So in this one, as I mentioned before, I am from the medical biotechnology, medical nanotechnology, nanomedicine area. So I'll be talking further, a little more about the medical area, the application of nanotechnology in biotechnology. So the first one, when you are talking about the medical application of nanotechnology, so first thing would be nanomedicine. So medical application of nanotechnology, which is less than 100 nanometer. So it comes into three major categories. One is nanodiagnostics. How this technology can facilitate early detection of diseases. Second one is the nanotherapeutics. And the third one is tissue engineering and regenerative medicine. So we'll see slightly in detail. So nanomedicine is nothing but nanotechnology. It's not only medicine. That's a point which I want to mention. When we hear the term nanomedicine, it's nanotechnology used for the treatment, diagnosis, monitoring, and control of biological systems. It includes delivery and targeting of pharmaceutical, therapeutic, and diagnostic agents using nanoparticles to cancer or other cells or other malignancies or other diseases. It includes nanomaterials used for regenerative medicine. This all comes under the major heading nanomedicine. So the potential applications: drug delivery, medical imaging, diagnosis, and tissue engineering. So before going to the details, let's have a quick look at the cellular kinetics. So human body contains a lot of cells. We all know that. Cells can either be non-dividing or it can be in the terminally differentiated stage. It can be continuously proliferating. And some may be recruited into the cell cycle. So when we talk about a cancer, a tumor becomes clinically detectable when there is a mass of 10 to the power 9 cells, nearly a gram. Until then, nobody is detecting it. It is not causing us a problem. No. And that patient is perfectly normal. But when the vascularization or angiogenesis is proper, when the cells can get necessary vasculature, the transition of this tumor mass from this size to the next stage is going to be very rapid. So generally, a detection of this malignancy is happening when it is moved from this stage to the next stage or in a later stage. That's a problem. So currently we have multiple detection techniques like PET, which is one of the powerful technology, positron emission tomography, and we have MRI, and we have CT, and we even have SPECT, right? But the minimum detectable limit of most efficient diagnostic tool today, that is the PET, is nearly 100,000 cells. But remember, this development of a cancer from the protein level to 100,000 cells takes nearly 10 to 15 years. It varies. Of course, it varies. In some cases, in really a malignancy, in virulent cases, in aggressive tumors, it may be faster. But in general, transition from a protein level to a mass of nearly 100,000 cells takes 10 to 15 years. So that means we miss 10 to 15 years for the earliest possible detection. As young biotechnologists, what we can do into this? So we can save a lot of life. So I'm not saying that we have, as people who are into the cutting edge nanotechnology area, we solve this issue. We are working in this area to see whether we can detect it early. So it's another pictorial representation of what I said in the early stage, because 10 to 15 years are quite a long time. If there is a tool, if we can detect it, there is some problem happening there, it would be much, much easier to destroy that tissue at that stage. But how can we detect? So we need innovations in the early detection of cancer for better cure. What could be the possibilities? Maybe a particle, a probe, which can go and specifically bind onto some of the markers of those cells, so that some of the imaging modality like PET or SPECT or even MRI can pick. So in our center, we have developed nano contrast agents like silica-based, iron oxide-based, calcium phosphate-based, quantum dot-based, but it still needs improvements. We have multiple publications and patents on this, but it's not enough for the early detection of cancer. So we need innovations in this area. So this is all what I was talking about. We have quantum dots, we have magnetic nanoparticles, we have gold nanoparticles, we have polymeric dots, These all can be functionalized with certain moieties which can specifically target the markers in the malignant cells, and that can be detected by certain imaging modalities. Then it will be an early detection. Now, from the detection, we can move to the therapy, what we can do. So conventional chemotherapy, the moment you think about chemotherapy, you would be seeing the undesired effects. Of course, it is giving the patient some relief. Maybe it is cured. At least they are extending their lifespan for a couple of years, 2-3 years. But the kind of undesired effects, it is happening to a patient's body. Its digestive system is gone, and the skin is gone, hair is gone. It's all happening in the conventional chemotherapy, but we have no other option. That's why we are trying to give this chemotherapy. So the conventional method is to give anti-cancer cytotoxic drugs to destroy cancer. One of the major problem is it is not specific. It will affect any cell which is getting in contact with these drugs. So these are the limitations: non-specificity, toxicity, adverse side effects, poor solubility. And this is the cytotoxic effect. And our delivery method, how we are giving this drug. Oral medications are there, inhalers are there, intravenous administration there, subcutaneous injection, infusion pumps, etc. are there. They are low cost, ease of administration, patient familiar, but still something is lacking. So this is what is happening. This is problems associated with the traditional drug delivery approach. When we are giving a particular concentration of drug, the drug which is reaching that particular malignancy or tumor is much lesser than the amount which you are giving. So in effect, for example, if a tumor is needed in some drugs in nanomolar concentrations, when you are giving to a patient, they have to give in milligrams of concentration so that some part of the drug is reaching that tumor. But what will happen to the rest of the drug? It will be degraded by the degradation mechanism or elimination mechanism in the body. It will cause toxicity to all the organs. So much of problems. But we have no other choice now. This is where innovations are necessary. So reduced potencies because of partial degradation, toxic levels of administration, increased costs associated with excess dosing. So as I said, for the treatment what we need is in nanomolar concentrations. But because when you are giving it to the entire body in kilogram, per kilogram, this much drug has to be given. So the cost will be too high. Compliance issue due to administrative pain. Is there a solution? So there is a solution, that is the controlled drug delivery. Controlled drug delivery is not only based on nanotechnology, but it can be delivered by multiple means. So what are the pros and cons? Pros will be achieve more effective therapies while eliminating the potential for both under and overdosing. Controlled delivery means we are giving effective dose in an effective way, not giving everything as a bulk. Maintenance of drug levels within the desired range, the need for fewer administrations because it can be sustained, optimal use of the drug, increased patient compliance. What are the cons? The possible toxicity or not. When we are delivering drug, it is not only drug, we are encapsulating this into some other delivering material, such as polymers. So the possible toxicity or non-compatibility of the material used, undesirable byproducts of the degradation, because the carrier will also be degraded, the chance of patient discomfort from the delivery device, high cost of controlled release systems, for example, the pump, infusion pumps, etc. So is there anything nanoscience can do in the delivery, drug delivery? Yes. For a nanotechnology, a biological cell is a large space to play with. It is in microns. When we see it in the microscope, we know it is nearly 5 micron to 10 micron, and when it is stretching, it can reach even up to 100 micron. Nanoparticles we are talking is about 100 nanometer. Remember, 1 micron is 1000 nanometer. So nano devices are small enough to enter cells. So you can target, a nanoparticle can specifically target some of the organelles inside it can easily enter the cell. It can intercalate with a certain mechanism inside the cell. So those possibilities are there with the nano devices. So how nano works for drug delivery? Nanotechnology will not replace, we have to remember that. Traditional thing is the pharmacology. Nanotechnology will not replace pharmacology, but will work with current pharmaceutical technology and improve delivery methods. So it can eliminate re-administration of treatments and use of implant that offer the same control over the overdose and over treatment cycles. It can just give a sustained release, more effective targeting of the drugs. The drugs encapsulated within a particle can be functionalized with a particular antibody, which will just go and bind into a cell types which has that particular receptor. And because of the enzymes in that particular location, the particle will be degraded and the drug will be delivered in that particular place. So that is possible. So new delivery modes, opportunities for controlled and sustained drug delivery will increase with the nanotechnology. So this is another slide which is just telling you what happens in the case of a nanoparticle. Because of their smaller size, nanoparticles are taken by cells, while large particles would be excluded or cleared from the body. That is important, because when you might be asking why it cannot be a microparticle, because the moment you take it in, I mean, you have to take it through some of the conventional route, either IV or even through oral route. The microparticles won't come to the circulation. It may be just eliminated through your fecus. If it is in the circulation,
Excuse me. Hello? Hello?
I think there is some connection loss.
Connection. Yeah, you have been muted, I think.
Is it? Probably you can call him.
Yeah, please unmute your Dr. Binulal. Hello?
Dr. Binulal, can you hear us?
yeah.
Yeah, now it's okay. Yeah.
I don't know what happened.
So maybe somebody has muted you.
Somebody muted me.
Yeah, I think. Okay, fine.
All right. So
Okay.
I think I talked. I mean, did I start? You were. Whether you were able to hear something from this slide?
Yes.
Yeah, started from
No,
yeah,
yeah.
I can start.
So I mean, this was basically, I was talking about what the size scale, how the size scale makes a difference. If it is microparticles, the saving mechanisms in our body can eliminate these microparticles from the circulation. Even if you are taking through the oral route, these microparticles won't be taken into circulation because it will be just pass out through the fecus. In the circulation, if it is IV, in the circulation, the saving mechanism like your liver, kidney, they won't allow these particles to stay in the circulation. We need the drug to be sustained in the circulation for long for a effective therapeutic, efficiency to get therapeutic effect. So these nanoparticles can easily escape these filtration mechanisms, and it can stay in the circulation for a prolonged time. It can go into the cell through the nanopores. So those are the advantages of these nanoparticles. Because of their small sizes, nanoparticles can be taken up by cells, while large particles would be excluded or cleared from the body. So if, as I said before, if you can, because now we understood that these nanoparticles, if you are given into IV, it can stay in the circulation for a prolonged time until the drug is degraded or the particle is degraded. Now think about, now the drug is inside a nanoparticle and the drug will be exposed to the enzymatic degradation only when the nanoparticle is degraded. Now if we can conjugate a particular marker, a particular ligand to the surface of this nanoparticle, and if it can go and bind a particular location where there is a malignancy, then the enzymes in that particular location will be degrading this nanoparticle so that the drug will be released, drug will be interacting with the cells, the malignant cells or disease cells, and a better therapy can be possible with this route. So this is another picture of the whole concept. See the cancer cell, and the nanoparticle has 2 dots there, and then they can interfere with the cancer-specific DNA because it can be complexed with the certain probes which can intercalate with the DNA, or it can be cancer-specific receptor. This particle can have, we all know that, biotechnologists know that when it is the malignant cells can express certain specific markers when it is turning into a malignant stage. So these particles can have these ligands, and it can be conjugated with another biomarker. It can be conjugated with a chemo drug. So a particle can be linked with so it can have multiple functionalities. It can have a biomarker. It can have a receptor for a particular cell surface receptor. So that when this is going and binding, the investigator can track whether it is going and binding there. It can have a particular drug. For example, doxorubicin is a drug which is highly used in the clinic. This can be loaded into this nanoparticle, and when it can be targeted to a cancer cell. When it is going inside the cell, it will be delivered, so that the effect will be much better. So as shown here, you can develop a nanoparticle like this one, and this combination delivers the same drug but with greater specificity, reduced doses, slow release, sustained release, and it can be loaded into an injectable therapy or as a patch. Think about loading this in a patch which will be released in a sustained way into the system. And this is the same traditional drugs. The color indicates the drug. When you're giving an IV, the drug is going to reach everywhere. But when you give as nanoparticles, of course, it will not be if it is not functionalized with a particular receptor, ligand, it won't be very specific, but the spreading is much confined here. So here, it's another pictorial representation which is telling you what is the difference in the traditional treatment. Even though drugs can kill cancer cells, it can kill non-cancerous cells as well, and there may be some of the non-cancerous cells escaped. That's why after the chemotherapeutic treatment, the patient is really weak. They will have a very damaged immune system, damaged digestive system, etc. But with a nanoparticle, nanotechnology-based treatment, this non-specific effect can be minimized. So it can kill specific cells which are very specific cancer cells, and but it can keep the non-cancerous cells alive. So this is a typical nanoparticle design where you have a drug inside, which you have a surface which is modified with your molecule of interest, and there is a targeting molecule. And then it can be multifunctional because it can target, it can deliver, it can report. So because it is in the early stages of its development, we need to know whether it is going to a particular location or not. So to summarize here, the drug delivery improves stability and solubility of drugs. For example, you all would be aware of this curcumin. Curcumin is highly insoluble. When you can encapsulate into a nanoparticle, its availability of this particular drug, potent drug, will be much, much higher when you are putting inside a particular nanoparticle. It increases the bioavailability of drugs. After certain pharmacokinetic of drugs, alters the pharmacokinetic of drugs. Example, it can extend its half-life, provides multifunctionality, targeting, delivery, reporting, etc. Provides improved therapeutic index, lowered toxic side effects, delivers multiple drugs directly into the tumor site. If you can load one drug, you can load multiple drugs. Now, we all have to remember that now the medicine is moving towards personalized medicine, because the same drug or the same combination of drug which is working in one patient is not working in the other patient. So now the medicine is moving towards a personalized approach, because each individual, the response rate for a particular drug for the same kind of a disease is different in different individuals. So when you are developing, you can have personalized combinations ready for a particular patient. and it enables nucleic acid delivery as well. And how this can be achieved? Nanosuspensions and nanocrystals, liposomes, solid lipid nanoparticles, nanotubes, nanowires, polymeric nanoparticles, etc. are the choices. And you have all these as pictorial representations here. And in our center for the delivery, we are using a lot of polymeric nanoparticles. And biodegradable polymeric nanoparticles are in the clinic, and there are products available in the market. And generally, even though a large variety of biodegradable polymers are available, based on the investigators' interest and the application and the particular application, how fast it has to be degraded, how small your nanoparticle has to be, we will be selecting the choice of our polymer, and we can tailor its properties. Because when we have two classes, I'm not talking into the details of the polymer technology, because we have biological polymers as well as synthetic polymers. The synthetic polymers now we can have with tailored properties, predictable, lot-to-lot uniformity, free from concern from immunogenicity. When you are taking a natural polymer, there will be immunogenicity, and we can get it from the reliable source of raw materials. And here I would just want to introduce you certain the nanomedicines, nanoparticle-based medicines, which are highly used nowadays in the clinic. For example, you might be, you know what it is? It is paclitaxel, which is a highly used drug loaded into albumin nanoparticles. They are making a lot of money. Because paclitaxel, as I talk to you, if you are giving IV for a breast cancer treatment, the patient is suffering from all sorts of side effects. The same drug, when encapsulated within a nanoparticle of albumin, it significantly reduces the side effects with the same efficacy. We have doxorubicin also as a chemotherapy agent that is coming out as Doxil, ambisome, which is amphotericin B, as an antifungal agent. So these examples are there in the market. Now, one more quick point which I want to make is idea which I want to cover is tissue engineering, because now what is tissue engineering or regenerative medicine, which is one of my favorite areas, because there is a huge demand for organ transplantation because of the lifestyle or many other stress conditions or the pollution. Many of the organ systems or organs or tissues are not working as it is required, and there is a very high demand for the transplantation. But there is a very obvious shortage in the organs. So that's why these technologists or engineers came out to see, can we develop tissues and organs in the lab? Now, still it is in the infancy, but later, maybe 25 years from now, we might be able to develop functional organs in the lab. So what is happening there? The concept is very simple. Like, we are trying to develop all these organs in the lab. That's the whole idea of this area. But a couple of tissue engineered products are available in the market. One is the skin. What happens here is we are trying to mix cells, for example, stem cells, which we can get from the donors or even from umbilical cord blood from the hospital. We can isolate mesenchymal stem cells and we can put it into a polymeric platform. Hope you remember the slide which I showed, where when the cells are put it into a nanofiber versus a microfiber, it behaved differently. So according to what we need, we can develop a scaffold made out of a polymer, and we can combine it, and we can allow them to grow in vitro in a bioreactor setup or in 2D culture, in the stationary culture, not 2D culture. 2D culture is an old concept where we used to culture cells in a 2D platform, where in a flask or a well, which is much, much different than how they are in the human body. That's why, even though we have amazing results in the in vitro, when we are trying to go into an in vivo study, we are getting just opposite results because we are testing the drug in a totally different platform. So, and try to develop tissues and organs from the lab. So these are the areas which I want to mention. And just to summarize, what nanotechnology can provide. So nanotechnology can be a cutting-edge technological platform to improve the process and products of all the areas of biotechnology for better output, better shelf life, improved efficacy and quality, reduced production cost, and reducing waste. I showed you examples of only medical side of it, healthcare side of it, as nanomedicines, and I just touched up on regenerative medicine or tissue engineering and drug delivery and early detection, diagnostics. But hope you all remember the areas which we discussed in the biotechnology. This technology can be used. For example, think about that. That is also in the medicine area. Think about a chewing gum which can sustainably deliver insulin. Why we have to go for taking injections? Now the challenges will come when the insulin is coming in contact with the digestive juice or to saliva. It might be losing its efficacy. Can you think of encapsulating this in a nanoparticle form? Even in the digestive juice or in the saliva, it won't be degraded, but it will be absorbed when it is reaching in the small intestine. Then we don't need any more injection. We just need to take a chewing gum. B means those who are suffering from diabetes. Think about improving the efficacy of a production by improving the surface area of a particular platform. For example, you want to sort something out and you want to conjugate beads which are very specific to a particular bioactive component. In the normal 2D surface, you would be able to, there will be a limitation to limitation in the surface area which you are available in your plant, in your production plant. But if you can think of designing a nanostructured, nanofibrous, or some other structured filter or a reactor, the amount of these capturing molecules which you can immobilize is multifold. You can increase the production in multiple times. So, but these young folks has to think how and where and when we have to use this technology. So before I finish off, I just want to introduce that in our center, we are having a master's program in MSc and MTech programs in nanobiotechnology, molecular medicine, and nanoscience and technology. MTech programs are for those who has master's degree, and MSc programs are with undergraduate, the graduates like BSc students. And this year, deadline is this September 19th. Those who are interested can contact the department if you are interested to know about this area, and if you want to focus, you want to learn this one. And these are some of the facilities which we have. We are one of the state-of-the-art facility for nanotechnology and nanobiotechnology. And with that, I just want to introduce some of our masters currently working as scientists in INST Mohali and IIT Hyderabad and in abroad. These are all our master students. They graduated from our center and they are doing great in the area of biotechnology and nanobiotechnology. That's what I wanted to tell you. With that, I want to stop here. Thank you all. And if you have any questions, please feel free to ask me.
Thank you so much, Dr. Binulal, for enlightening us with the world of nanobiotechnology. And it was a very informative presentation, as we could see from the chat box. Many have thanked you for such a crisp and clear presentation that you have made. And we are also having a lot of questions to ask for you. Let's start that.
Yeah. Please go ahead.
Okay, fine. One question is from Anil Menon. He's asking, what is the role of nanotech in regenerative medicine?
It's a good question. I mean, maybe the best question which I can answer. All right. So regenerative medicine as such, it's a broad area, just like what we discussed, the biotechnology. Regenerative medicine is trying to regenerate any deformative tissues or organs, right? For example, if a particular part in the liver is not functioning as it is required. So there are multiple ways which we can do it. So there can be a liver transplantation. That is what is happening nowadays. There should be a donor. So in regenerative medicine, tissue engineering can be one aspect. Now, tissue engineering means you are trying to engineer functional tissue ex vivo. So now you have multiple choices, right? Platform where your stem cells can be cultured. You can definitely select a micro scale scaffold or hydrogel, but you can have a nanoscale platform, because I have shown you one example of how the cells behave differently when you give a different topography. So you can have a nanostructured scaffolding system to culture cells which are capable of producing functional liver-like tissues or liver tissues ex vivo. That's one application. That's where you can apply nanotechnology. Second one, if you want to deliver certain drugs, for example, in the case of bone, there is a defect in osteoporosis. Okay, you want to deliver certain drugs to this patient with certain specificity. You can always encapsulate nanoparticles which can deliver your drug of interest. For example, BMP2, bone morphogenic protein. Okay, if you just inject bone morphogenic proteins, what can happen there is anabolism or catabolism. For example, when you give these bioactive molecules in supraphysiological concentrations, when we inject, there is no control, we are just injecting. That is supraphysiological concentration, but never in our body, these bioactive factors are in supraphysiological concentration. So we have to control the amount of this active factor available in the site of regeneration in a precise manner. In that aspect, nanotechnology can be one of the best technologies which you can use. Hope you understood what I wanted to convey.
Okay, the next question is regarding can nanotechnology be applied for RDNA technology? Is that having any relation with that?
Yeah, so I kind of, I think I actually thought about mentioning the RDNA technology and molecular biology. See, because I mean, to all the listeners, I want to mention this. We are having, when students are coming here for interview, okay, the question, one of the short questions which we ask them is, what you are interested in, what you wanted to do. So the answer is, they want to do molecular biology, they want to do RDNA technology. These are just technologies. They want to say what exactly they want to do. For example, if somebody is going to come and tell that they are interested to do nanotechnology, what nanotechnology they wanted to do, the question is very specific, right? Where you can use nanotechnology in RDNA technology. So, of course, you can use now the CRISPR-Cas technology, which is highly used in the RDNA area. They are using nano-based vectors for this particular purpose. And other than that, if the question can be in the particular RDNA, what exactly they wanted to know, what exactly they wanted to do, if that can be clarified, then it can be discussed further. Because nanotechnology is giving you an opportunity to manipulate things into a nanoscale, of course. It depends on, in the RDNA technology, what exactly they wanted to do. Whether they just want to edit a gene, or they wanted to do something further, they want to introduce a gene. So it depends on what exactly they wanted to do. There is a room for doing it. But I may not be the expert to do that, because I'm not a recombinant DNA technology expert. But in the center, there are experts who are doing it.
Okay, fine. What about its role, its applications in plant biology? Himanshu is asking.
One of the applications which I know is in the area of fertilizers. Plant molecular biology, I'm not very sure. Of course, that also comes in the RDNA technology, right? In the plant biology, because a lot of interests are coming out from because in a country like us, we need to improve our crop. We have to control the pest, etc. In there, now enormous amount of pesticide application and fertilizer application is happening, which is destroying the flora, the natural flora in the soil. But there, if you can have a nanotechnology based approach, where you may not need to apply certain fertilizers in this particular concentration to the plants or to the crop or to the soil, can be very much controlled using nanotechnology based approaches. It can be particles, for example, calcium phosphate particles coated with a certain this can be produced in bulk. Okay, calcium phosphate particles, calcium sulfate particles coated with a certain pesticide, active ingredients of the pesticides. Those can significantly improve the outcome. For example, in much lower concentrations, you can get the same effect what you are seeing by applying bulk amount of these fertilizers and pesticides. So that is one of the potential area, and a lot of studies are going. It's a very interesting and important area for our country. So other than that, in the molecular biology, of course, there are ways, but it depends on exactly what you wanted to do.
Yeah, there are similar other questions, whether to the applications in microbiology, in the AIDS treatment, in gene therapy. So
yeah, nanoparticle-based gene delivery delivery systems are a big established area. Another major area in the microbiology, because we are in a hospital, we have that multi-drug resistant bacterial infections. So when these particles, as I mentioned in the talk, when they are in the nano dimension, for example, metal oxides, for example, copper nanoparticles, zinc oxide nanoparticles, they are now used to coat the catheters, urinary tubes, and even the operation theaters and these surgical equipments, etc. So that the biofilm formation from these I am talking only about the pathogens. Okay, pathogenic biofilm formation can be controlled significantly. The other question is about the virus. I think AIDS or something. It was
AIDS treatment.
Against the viruses, now one challenge is which we are facing is, for example, we wanted to test a couple of things against COVID-19, okay, in our center, but we are not allowed to do that because the facility, the specifications which is required to do that is not currently available, because there are these zinc oxide particles and all. When you treat it with bacteria, they're just dying off. Whereas it is not very toxic to human cells. So in the bacteria, we have tested it. In the virus, currently, to be honest, we haven't tested it, but we know that there are certain aspects, because we can go as small as 5 nanometers in particles, which is much smaller than a virus. So that will have an inhibitory effect or a detrimental effect to this virus. But we currently, there are only National Institute of Virology. They are allowed to do this kind of testing, and they are overwhelmed with these kind of tests, other more clinical type of tests currently going on. So at present, we are not in a position to test it, but for sure, it will have applications.
Also, Justina is asking, is there any possibility that the nanoparticles used for therapy be considered by our immune system as an antigen?
That's one of the advantage of the nano system. That is very true if you are using a microparticle, because it can, again, it depends, okay? Again, it depends on what type of nanoparticle you are using. If you are using an inert material, because the immune responses happens because of the two reasons, mainly of two reasons. Because it is detecting the antigen, a particular epitope there, antigen, or it is just a size, okay? The size scale effect can be very much eliminated by using nanoparticles. It can very much escape the nano system, the immune system. For example, macrophages. That's what one of the areas which I'm working is in the immunomolecular properties of it. The macrophages can stay in two polarization states. One is a inflammatory stage where it will just give for example, if there is a cut, you will see the swelling, and it's in an inflammatory stage. The macrophages are infiltrate there, and they are just guarding there. And the other one is a regenerative one. There, they will tell, these macrophages will produce certain factors and tell the surrounding cells to go into a regenerative phenotype. They just ask, okay, you produce new tissue there. Okay. Now, when you are putting these macrophages into a nanoscale sized particles or scaffolds, compared to a microscale particles or scaffolds, the behavior is totally different. Okay. So the bottom line is, nanosystems has an upper hand in bypassing the immune system, mainly because of the size scale. But if the degradation products, for example, if it is based on the specific antigen, maybe there will be an immune response, but it can be much smaller, much milder than that of a microscale systems.
Okay, here's a question from Binoy Kurian. Can nanotechnology and artificial intelligence can collaboratively applied in medical field?
Tough question for me. Of course, I'm not very informed about that. I just know artificial intelligence.
Yeah. Okay.
Maybe a possible application will be there, but in the center we haven't, because our other campuses, for example, the Coimbatore campus, they are working in the artificial intelligence. Possibly, we might collaborate with them if there is an application like that. I can't think anything at the moment. I mean, for sure, you saw what is the possibility, right? And in the artificial intelligence, in the medical field, it's going to have a big impact in the medical field for sure. Now, how it can be linked has to be thought through. I haven't thought in that aspect.
Okay, fine. Then one Subhadeep Das is asking, how nanoparticles can be designed to be degraded at a particular location of target to release its drug?
How nanoparticles can be designed
to be degraded at a particular target, like after its drug delivery.
Yeah. So there are a couple of options. Okay. So even though we are talking about nanoparticles can be, as I told you, it can be less than 100 nanometer, but it can span from 1 nanometer to even 999 is nanometer. Okay, because 1 microparticle means it should have 1000 nanometers. Now, as a bulk particle, the bulkiness of the particle is one aspect which is going to influence the degradation. That's one. Maybe if you want something to be quickly released, it has to be as small as possible. That's one thing which I would suggest. Second one is more important thing is the material, the material which you are going to use as the particle. So degradation can be, for example, if you are selecting a polymer degradation can be enzymatic or hydrolytic. Okay. For example, if you are taking a polymer, for example, something which is responsible for enzymatic degradation, I'm sure that in the system it will be degraded much faster. But for example, polycaprolactone, the enzymatic action is very less. It cannot degrade enzymatically. But hydrolytic degradation can happen. That's why, for example, if you want to have something which has to be stayed there for a quite long time, in months, I would suggest you should select something which is someone else is presenting, I guess.
Yeah, no problem.
Okay. We should select something which won't be, sensitive for enzymatic degradation. But on the other hand, if you want something to be degraded, again, you have to know what all enzyme going to be present in the area where you are going to apply this nanoparticle. So accordingly, you can modify. Even the extent of cross-linking, especially in the polymeric nanoparticle. If you cross-link extensively, the degradation will be slower. If you are not cross-linking, the degradation will be faster. So there are multiple ways we can control the degradation of nanoparticles in a particular site.
Okay. Asking, is nanotechnology safe? Also, some other is having a question like, what substance can be used instead of harmful substances like lead and cadmium for nanoparticle formulation?
One of the widely accepted particle is gold nanoparticles. Gold nanoparticles are not known to be toxic, but if there is a material which is toxic, when it is in the nano formulation, its toxicity is going to be much, much higher. That's what I can say. That's for example, if you are cadmium itself is toxic. When you are taking into the nanoparticles, its effect will be much higher because the mechanism of action is in that way. It can go into. So toxicology, nanotoxicology is a major area where regulatories are carefully monitoring what is happening, and it has to be studied well. For example, zinc oxide, those particles are silver nanoparticles. They're relatively safe, but depends on, for example, silver has a very high antimicrobial effect. Okay, so now you are using silver, and if you are just throwing that, silver containing thing, for example, you are using silver in a washing machine, silver nanoparticle, and if you are draining it, it will kill all the microbes in there in the soil as well. So there should be some way to reprocess it, to eliminate this. When we are in the drainage, it should not go to the drainage. So there are controlled ways where we can exploit the benefits of nanotechnology, where in the same time we can prevent its harmful effects. It should not be toxic to the society and to the environment. So it has to be in a balanced manner. That's what I would say.
A few other.
You ask something more? That was the question, right?
Yeah. Is it safe that you have answered? No?
Safe, it depends on what nanoparticle you are taking. Okay. For example, nowadays everyone is knowing that asbestos. It's kind of being inhibited everywhere, because these fibers are gonna go into the lungs, and it's gonna cause problems in the long run. But again, it depends on what nanoparticles, what concentration, what material it is, how it is taken up, how frequently you are taking, because we are exposed to nanoparticles in our daily life as dust or some other particles, as fumes or as pollens. Those are microparticles, but when it is in the degraded form, it is. So anything too much can be harmful. That's why, so I'm saying, right? I mean, for example, the same paclitaxel, a drug which is a lifesaver, when it is given in a bulk, it is killing all the other systems. But when we can control it, it is giving us beneficial effects. So it's not a blind technology. It cannot be used in a blind way. It has to be in a controlled way. So our regulatories are looking into it. There should be a regulatory use for in any aspect, whether it is nanotechnology or any other technology, even biotechnology. If you are using biotechnology, you are genetic engineering in a faulty way, of course, you can produce pathogens which cannot be much more pathogenic than your COVID-19, right? So there should be some controlled way of dealing it.
Okay, thank you. There are a few questions regarding the nanotechnology and cancer treatment. So one is, Soumya is asking, is nanotechnology treatment for cancer affordable to normal people? Is it costlier than the conventional treatment now carried out for cancer?
Good question. It's a good question. At the moment, because there are two aspects to it. Okay. For example, there are certain nano-based nanomedicines which are very expensive. Those are targeted drug delivery systems. For example, you have drugs which are targeted by a monoclonal antibody to a tumor. Okay? At the moment, it is very expensive because the companies are invested a huge amount of money to make it, and slowly it will come down. But your question, whether it is affordable, you have to see it in two ways. For example, the amount of drug you might need to go. If nanotechnology, for example, a single dose of 100 nanomolar concentration is good enough for the entire treatment, that might be balancing the amount of money which is required for multiple cycles of chemotherapy and associated treatment and the side effects treatment, etc. So at the moment, it might sound as it's expensive because the Abraxane, the paclitaxel may not be expensive. Compared to paclitaxel, the nano formulation of the paclitaxel is expensive. But now, including our center, we are trying to see alternatives. That's why we need the cost will come down when you young folks are coming into the science, and you are producing it in our country. Now this is something which is produced some other country, and they are giving us this much cost. We have to afford it. We have to buy it. So that's why government of India is giving us opportunity to come up and and contribute. That's why I use this word many times in the slides, in the presentation. We are expecting for your contribution. When it is made in our country, the cost is going to come down significantly.
Yes, Archa has a doubt, like, is there any chance that these nanoparticles can cause cancer?
These nanoparticle is a very generalized term. What nanoparticle and why, I don't know. Nanoparticle as such, we are not, these are, when we are making it in a controlled environment. Even the investigator is not supposed to expose to the synthesis of nanoparticle. For example, if I am the fibers which I showed near to the hair SEM, those are routinely, because I'm making those nanofibers for the last 15 years. Okay, but I'm using it in a controlled environment where it is inside a biosafety cabinet. It's inside a chemical cabinet, chemical hood, where it is connected to a vacuum, to an exhaust, where it is not coming and getting exposed to the investigator or the person who is actively engaged in research. The problem there is the common people may not be getting exposed to these things, but a person who is continuously doing research in a particular nanoparticle or a nanofiber or a nano application may get exposed to the same thing in a prolonged way, right? There it may have a negative impact. Therefore, all this research, that's why the facilities, I quickly showed the facilities. We have glove boxes. I mean, this is not a technology where we can do something on the bench and you just mix and see it like this one. A controlled way, and if you are using in a controlled manner, I think it's safe. It depends on the what type of material you are dealing with. Nothing it can be just used freely. Even the drug, for example, paclitaxel, it's a lifesaver. You think it can be just exposed? It can be inhaled? No, it cannot. Even curcumin. Curcumin is very good, but if you are continuously exposing to curcumin, I'm sure that your lungs will have problems.
Yeah, which is better, that the chemotherapy or nano-based therapy is better?
For sure, I would recommend for a therapy which is more specific. Okay, I would say more specific. If there is some other way to be more specific than nanotechnology, right? Because nanotechnology is one way which I know to be more specific than the bulk treatment, but currently that is not that much developed, right? So the affordability of a nano-based formulation to a common man is not that easy. So they go for the bulk chemotherapy. Always it is good. Think about this scenario. You have a problem. You have a cut in your hand, in the fingertip. Okay? You are putting Bernal to the entire body. That is what is happening. Okay? So can we have more specificity in the treatment? So that's where in the medical area we are trying to see.
Okay. Regarding the COVID-19 treatment, is there any nanomedicine nanoparticle which is found to be promising against?
At the moment, so what we are trying to see is we are interested in the capturing of this particular virus. For example, if it is, everyone is wearing mask. I also have a mask near to me because I'm sitting alone in the room. It is okay at the moment, but currently there is nothing. Even reports are saying that it can stay in a surface for quite prolonged time, right? In a quite, prolonged duration. So even if it is coming in the environment and it is coming into the mask, it is not being killed. You are taking it, you are touching it, you are putting somewhere else. So is there a way which can be antiviral in the mask, in the material at the moment? That's one research which is currently going on. Another one is nano vaccines. Nano vaccine is, I don't know how many of you are familiar with the nano vaccine. Currently, it's mRNA vaccine is coming up as a very promising area. And mRNA vaccines are in the nano formulations, it is coming out as nano formulations, but at the moment, I'm not aware that some nano-based systems are developed against COVID-19. I'm not aware of that because it is too early to say something like that. But eventually, something will come out. The one thing which I can say is nanofibers are very good filters for this COVID, because the pore size which we are having is less than 100 nanometer. So it cannot pass through if you are wearing it. So we are having an N96 mask. N96 is a fiber which can prevent more than 96% of particles coming in this nano size. So as a drug, I'm not aware of that at the moment. But those are areas where these young folks can, focus on.
Binoy Kurin is again asking, can you say some specific examples of nanomedicine and corresponding treatment currently followed in Amritha Healthcare?
Amritha Healthcare in the hospital?
Yeah. It has got some applications in hospital settings.
Yeah, of course. Abraxane is commonly used. It's a paclitaxel encapsulated within the albumin, and some of the monoclonal antibody-based one. I mean, we are using in the hospital, but it depends on the in the affordability of the patient. Of course, it is expensive, especially when it is in the monoclonal-based one. Rituximab, if I remember right, because it's in the clinical side, we are in the research side.
Okay. A very basic question, very general question from Gauri. How can we enter and proceed a career in nanotech after completing bachelor's degree in biotech? And how much is the field prominent in India?
Okay, I think in the webinar series itself, I think you had a couple of series on So in the nanotechnology as such, see, the field cannot be just isolated from everything else, and it is in combination with many other things. After finishing your bachelor's, see, I basically showed you multiple areas where biotechnology, we need government, India need contributions and developments and technologies and products, right? And we are using this technology, nanotechnology, as a promising thing to enable or enhance in that. So in that aspect, for example, if there are, 10 players, think about this scenario where you want to go for a long jump. Okay, that's how I put it. And we want to represent somebody, we want to hire somebody to our center who is going to be the best in the long jump. We'll always see who is well equipped for the long jumping. Is that right? So the person who is well equipped will be selected first. Of course, when the opportunities goes up, we might need to select everyone to produce. But at the moment, we are looking for the industry or in academia, we would be looking for a person who has some experience, better understanding compared to others. So what that particular student can do is they can look for studying it further. If any career, in any area, people can go for two streams. One is industry or in academia, in research and development, right? If you are in industry or in research and development, of course, you have to go for a postgraduate degree. After postgraduate degree, you can decide that whether you want to go for a doctoral degree and training in India or abroad. With that, you will know which area you want to focus, how things has to be investigated, how things has to be conducted. In industry, after a master's degree, I think in nanotechnology as such in India, there may not be a nanotech industry. Maybe there are some carbon nanotube, which is not in the biotechnology area. But those pharmaceutical industry and the startups which are currently happening, which are promoted, are currently looking for people who are trained in the latest technologies. One more thing which I want to add is, now Government of India, through various schemes, they are facilitating R&D very much. There is some scheme called Big Innovation Grant. Anybody, even you are a 10th standard student, you can apply if you have an idea. It doesn't need, that's how the government is planning now. But if you have a higher degree and facility, it's well and good. You don't need. So if you have an idea, if you know what you wanted to do, if you identify your niche area and your interest, there are options. What is required is your interest and how dedicated you are.
Yeah. And can you say something about organ 3D printing, which Aditya is asking?
Okay, so I couldn't include that. I mean, these are my specific areas. Organ 3D printing is at its infancy at the moment. We can print models. I mean, if somebody is telling you that they are printing organs, don't believe them. Okay, at the moment, because we have one of the state-of-the-art printer, like from the 3D bioplotter. This is one of the best printers which can do 3D bioprinting. 3D printing is mainly two streams. Okay, in our hospital, we are 3D printing a lot. That is model. For example, the clinicians, before they go for a heart surgery, from the CT, they 3D print an organ. And these doctors, they actually physically try, in complicated cases, they will try the surgery before they operate the patient. That's where it is happening. But as a functional organ, it is too complicated at the stage. What happens is, even in our center, what is happening is, now we are trying to develop organoids for drug testing. Organoids for drug testing is a major area at the moment. But eventually, we might be coming out with a small vessel, vasculatures, blood vessels. Those are simple things which we can try. But organoids is growing up as a big area because, as I quickly mentioned somewhere in the talk, the testing which is currently being done in 2D cultures is not actually recapitulating the in vivo environment of diseases or functional organs. So organoids is miniatures of these functional organs or disease condition. For example, in one of our project is 3D print and or model for prostate cancer to test drugs and drug combinations. So that's an active area at the moment. But eventually we will progress, because 10 years, 20, 25 years back, we never thought we will be in such a situation, such a technologically advanced stage now. But so similarly, we will very much progress towards the organ printing, but not at the moment. Currently, if you are interested in the organ, decellularized organ systems, recellularized, decellularized cadaver organs, recellularized is one of the potential areas because they have certain applications, but it's always donor, donated organs are currently being used in the clinic. And another, even in the stem cell, there's only one approved stem cell transplantation in India. You know that it is hematopoietic stem cell. All others are just trials and tests.
Okay, one last question. Question by Archa. Is there any chance of the cells getting mutated by administering the nano-based drugs?
Come again, I'm sorry.
Yeah, is there any chance of the cells getting mutated when we administer the nano-based drugs?
I'm not in a position to say no, okay, because we don't. Mutation, again, in the analysis, for example, the genotyping and all which we are doing currently is not reflecting anything. Because you are in this nanotechnology based one, for example, if you are in the therapy, you are trying to kill already mutated cell. That's a malignant cell. So exactly in the genomic level and in the mutation level, what exactly is happening is not known. But if we are worried about this one as a problem, whether it is getting mutated or not, I think we are in a much difficult situation. For example, I'm sure that when you are trying to stake cells and when cells which lived in a three-dimensional environment in a totally different area, when you're trying to culture in a plastic, which is really stiff, they might be mutating. They might get mutated. That's why the regulatories are not allowing for culture expanded cells for implantation. The clinicians are allowed to take the cells and put it if they are not expanded in a petri dish. But the moment we do expansion, the regulators regulatories are stopping. They are stopping us, say that, no, we need further studies. We can't use it in the patient. So that one, again, it cannot be generalized. What nanoparticle, what concentration, what frequency is going to make a mutation, whether it is making. That has to be studied extensively.
Okay, with that, I hope all the participants are having a very nice time listening to you. There was a very good interaction, and you have answered almost all the questions raised by them. And with that, let's conclude. On behalf of Kerala State Council for Science, Technology and Environment and Kerala Biotechnology Commission, I extend a very thanks to you for having spared your time. Almost half an hour you have been interacting with the participants, and I think I also thank all the participants for this wonderful interaction. And Dr. Arunan, sir, do you have something to say, sir?
Yeah, actually, it was wonderful, Dr. Binulal. Actually,
Thank you.
Yeah, you actually shown the vast experience, your experience to the participants in nanotechnology. I'm sure that most of these participants actually have benefited out of this, and I'm sure they will find better opportunities by this talk. And,
Thank you. I also thank you, the Kerala KSCSTE, because I got a very enthusiastic, because I'm seeing like more than so many questions and so many questions coming. So what we need is this kind of energetic people, enthusiastic people. I enjoyed it, and thank you for giving me an opportunity to interact with these kids.
Thank you, Dr. Binu.
Thank you.
Thank you.
Thank you. All right.
Thank you.